So how was the field trip today? Did you all have fun? What did you guys think?
It was fun seeing all the pollinators at work, but there's something I don't understand.
And what's that?
Well, I understand pollination and fertilization and how they work, but what is pollen tube attraction? You mentioned the pollen tube, but I just got confused.
As you know, our job as pollinators is to help carry a pollen from one flower onto the stigma of another flower. That alone doesn't mean the plant will be fertilized. A pollen tube has to be formed. The tube is a polarized cell that is germinated from a pollen grain and has tip growth that is similar to how a plant's root hairs grow. As a tube grows through the pistil of the plant, the pollen tube acts as a straw that the sperm will travel through down to the ovule. When the tube reaches the ovule, it invades the synergid cell and ruptures, discharging its contents. This allows the sperm and the egg cells to meet, and double fertilization occurs to produce the endosperm and zygote.
How does pollen tube attraction work?
Well, to start, the female tissue communicates with the pollen tube to assist in its journey to the ovule. If a pollen grain is incompatible, it will be rejected. If accepted, the process begins. The entire process can be divided into two parts that focus on communication: the pre-ovular and ovular guidance. Pre-ovular guidance is the navigation of any compatible pollen tube from the stigma to the ovary. The transmitting tract constitutes the path from the stigma to the ovary, where the tube interacts with guidance signals. Multiple proteins are responsible for the tract development, such as HECT1 and chlorophyll proteins. Before getting to the ovule, the pollen tube exits the tract in what is known as pollen tube emergence. The pollen tube continues its tip growth until it reaches the ovule. Not much is known about the process of emergence other than two family genes, CHX21 and CHX23, are expressed in the pollen tube and assist with the directional growth through regulating pH. Following emergence is ovular guidance.
Ovular guidance refers to the precise navigation of a particular pollen tube to one ovule. It can be split into funicular and micropillar guidance. Funicular guidance is a mechanism of pollen tube guidance from the surface of the septum to the funiculus. The funiculus is a stalk-like structure that connects ovules to the placenta and the ovary. Micropillar guidance is a mechanism of pollen tube guidance from the funiculus to the micropyle. The micropyle is a small hole that is formed without closing the edge of the integument in the ovule. The pollen tube penetrates the embryo sac through this hole. Pollen tube attractants are active in the micropillar guidance.
Multiple pollen tube attractants have been found in various plant species. The first reported one was ZMEA1, or Zay maize egg apparatus 1, a peptide exclusively expressed in the maize egg apparatus before fertilization. One experiment found that downregulation of this peptide expression resulted in misguidance of pollen tubes to the ovule and fertilization failure. Therefore, CMEA1 was considered as a signaling molecule for short-range pollen tube guidance.
A common attractant found in plants are the lures. In T. fournieri, TF lure 1 and TF lure 2 are defensin-like CRPs and are pollen tube attractants that are responsible for micropillar guidance. They are small peptides and are predominantly expressed in the synergid cells. When recombined to gelatin beads, TF lures were still able to attract pollen tubes despite low amounts of TF lure 2 present in the beads. This showed that lure was still sufficient enough to give rise to pollen tube attraction. An experiment found that the downregulation of lure genes by injection of morpholino antisense oligomers into the embryo sac cells strongly decreased the attraction frequency of pollen tubes by the ovule. Lures belong to the most strongly expressed genes in the synergid cells. The encoded peptides are secreted towards the filiform apparatus and likely generate the necessary concentration gradient around the micropyle, which is important for pollen tube attraction and guidance towards the egg apparatus.
In Arabidopsis, receptors for lure were identified as a few receptor-like kinases on the pollen tube, like male discoverer 1 or MDIS1, MDIS1 interacting receptor-like kinase, MIK1, MIK2, and pollen receptor-like kinase 6, PRK6. MDIS1, MIK1, and MIK2 were found to localize to the plasma membrane, and knockout mutants for these genes show defective micropylar guidance. PRK6 localizes to the plasma membrane of the pollen tube tip, and this localization changes in an asymmetric fashion towards LOR before the pollen tube tip growth direction turns. Although pollen tube receptors for LORs have been identified, the full picture of pollen tube guidance is still unclear, and further analysis of the signaling mechanisms is needed.
So, why should we care about pollen tube attraction? Why is it so important?
Pollen tube formation occurs in all seed plants, including angiosperms. Pollen tube attraction is an important process because the pollen grain itself is non-motile and the egg cells are embedded deep inside the female tissues of the plant. Without pollen tube attraction, fertilization between the sperm and egg is difficult and can fail as the two are unable to meet. Thus, it is important to overcome this barrier in order for successful fertilization and the production of the next generation of plants.
In some plants, there is a single ovule, while in others there can be hundreds, so guidance and attraction is key for one pollen tube to meet one ovule.
Wow!
That's awesome.
I can't wait to start working as a pollinator.